toxicity with its only substrate being C4 and C2. Once C1 is activated, the recognition stage ends. Now we're going to discuss the activation unit. Phase 2, the formation of the activation unit results in the production of an enzyme known as C5 convertase. C4 is the second most abundant complement protein with a serum concentration of approximately 600 microgram per milliliter. It consists of 3 polypeptide chains and has a molecular weight of approximately 198,000. C1s cleaves C4 to split off a 77 amino acid fragment called C4a. In the process, it opens a thioester containing active sites on the remaining part, C4b. The C4b must bind to protein or carbohydrates within a few seconds, or it will react with water molecules to form a IC4b, which is rapidly degraded. Thus, C4b binds mainly to antigen in clusters that are within a 40 nanometer radius of C1. This represents the first amplification steps in the cascade, because for every one C1 attached, approximately 30 molecules of C4 are split and attached. C2 is the next component to be activated. Complement proteins were named as they were isolated before the sequence of activation was known. Hence, the irregularity in the numbering system, C2 is a single-chain glycoprotein with a molecular weight of 102,000. The C2 gene is closely associated with a gene for factor B or alternative pathway on chromosome 6. In the major histocompatibility complex, and each serve as a similar purpose in its particular pathway. When combined with C4b in the presence of magnesium ions, C2 is cleaved by C1 to form C2a, which has a molecular weight of 70,000, and C2b, which has a molecular weight of 34,000. This is the only case for the designation alpha to be given to the cleavage piece with enzyme activity. Enzyme C2 must be within a 60 nanometer radius of bound CLS for cleavage to occur. Binding of C2 to C4b can occur in the fluid phase, but C4b attached to antigen is much more efficient in accepting C2. This serves to keep the reaction localized. The combination of C4b and C2a is known as C3 convertase. This is written as C4b2a to indicate the complex in an active enzyme. This complex is not very stable. The half-life is estimated to be between 15 seconds and 3 minutes. So, C3 must be bound quickly. If binding does occur, C3 is cleaved into two parts, C3a and C3b. C3 is the major constituent of the complement system and is present in the plasma at a concentration of 1,200 micrograms per milliliter. It serves as the pivotal point for all three pathways, and cleavage of C3 to C3b represent the most significant step in the entire process of complement activation. The molecule has a molecular weight of 190,000 and it consists of 2 polypeptide chains, the alpha and the beta. The alpha chain contains a highly reactive thioester group, and when C3a is removed by cleavage of a single bond in the alpha chain, the thioester is exposed. The remaining piece, C3b, is then capable of binding to hydroxyl group on carbohydrates and protein in the immediate vicinity. Only a small percentage of cleaved C3 molecules bind to antigen. Most are hydrolyzed by water molecule and decay in the fluid phase. C3b is estimated to have a half-life of 60 microseconds if not bound to antigen. The cleavage of C3 represents a second and major amplification process because about 200 molecules are split for every molecule of C4B2A. In addition to being required for the formation of the membrane attack complex, C3b also serves as a powerful opsonin. Macrophages have specific receptor for it, discussed later in the chapter. And this contributes greatly to the process of phagocytosis. However, a large number of molecules are needed for this to occur, hence the need for amplification. If C3b is bound within 40 nanometers of the C4b2a, then this creates a new enzyme known as C5 convertase. Depicts this last step in the formation of the activation unit. The cleaving of C5 with deposition of C5b at another site on the cell membrane constitutes the beginning of the membrane attack complex or MAC. Now let's talk about the membrane attack complex. The C5 consists of two polypeptide chain, the alpha and the beta, which are linked by disulfide bond to form a molecule with a molecular weight of about 190,000. C5 convertase consisting of C4, B2, B3b splits off a 74 amino acid pieces known as C5a and C5b attached to the cell membrane, forming the beginning of the MAC. The splitting of C5 and the cleavage of C3 represents the most significant biological consequence of the complement system, as explained in the biological manifestation of complement activation section. However, C5b is extremely labile and it is rapidly inactivated unless binding to C6 occurs. Subsequent binding involves C6, C7, C8, and C9. None of these proteins have enzymatic activity and they are all present in much smaller amounts in serum than the preceding components. C6 and C7 each have molecular weight of approximately 110,000 and both have similar physical and chemical properties. C8 is made up of 3 dissimilar chains joined by disulfide bond and has a total molecular weight of 150,000 squared. C9 is a single polypeptide chain with a molecular weight of 70,000. The carboxy terminal N is hydrophobic, while the amino terminal N is hydrophilic. The hydrophobic part serves to anchor the MAC within the target membrane. Formation of the membrane attack unit is pictured in figure 6-4. Membrane damage is caused by at least 2 different mechanisms: channel formation and the binding of phospholipids. The latter cause a reordering and reorientation of molecules that results in leaky patches. When complement proteins are bound, membrane phospholipids rearrange themselves into domains surrounding the C5b6-7-8-9 complex, and the integrity of the membrane is destroyed. Ions then are able to pass freely out of the cell. The membrane attack unit begins when C6 binds to C5b, thereby stabilizing it. Then this complex attaches to the cell surface. C7 bind next, forming a trimolecular complex that has a high affinity for lipid constituents of the cell membrane. This allows for insertion of the C7, part of the C5b6-7 complex, into the membrane of the target cell. Next, C8 binds to C7 and exposes a hydrophobic region that interacts with the cell membrane to form a small hole in the membrane as C8 is inserted into the lipid bilayer. Lysis can be observed at this stage, but the addition of C9 accelerates the process. Binding of C8 causes a loss of potassium from the cell, which is followed by leakage of amino acid and peptides. The pore formed by the binding of C5b678 is small, capable of lysing erythrocytes but not nucleated cells. For lysis of nucleated cells to be achieved, C9 must bind to the complex. When C9 binds to the C5C8 complex, it unfolds, becomes inserted into the lipid bilayer, and polymerizes. Anywhere from 1 to 18 molecules of C9 can bind to one C8 molecule. C9 polymerize only when bound, and it is believed that the C5C8 complex acts as a catalyst to enhance the rate of reaction. Polymerized C9 forms a hollow, thin-walled cylinder which can be seen on electron microscopy and constitute the transmembrane channel. The complement MAC unit has a functional pore size of 70 to 100 A. One such unit can lyse erythrocytes, but lysis of nucleated cell is a multi-hit phenomenon. It was originally believed that lysis of all cells was a single-hit phenomenon. Destruction of target cells actually occurs through an influx of water and corresponding loss of electrolyte. Now we're going to discuss about the alternative pathway. Now, pathogens can be destroyed in the absence of antibody by means of the alternative pathway, which act as part of innate or natural immunity. This pathway is important as an early defense against pathogens. Phylogenetically, this represents the oldest of the C3 activating pathway, first described by Pillemer and his associated in the early 1950s. The pathway was originally named for the protein properdin, a constituent of normal serum with a concentration of approximately 5 to 15 micrograms per milliliter. Now, it is known that properdin does not initiate this pathway but rather stabilize the C3 convertase formed from activation of other factors. In addition to properdin, the serum protein that are unique to this pathway includes factor B and factor D. C1, C2, and C4 found in the classical pathway are not used at all in this system. C3, however, is a key component of both pathways. The alternative pathways is summarized in figure 6-5. Triggering substance for the alternative pathway includes bacterial cell wall, especially those containing lipopolysaccharide. Fungal cell wall, yeast, viruses, virally infected cells, tumor cell lines, and some parasites, especially trypanosomes. All of these can serve as sites for binding the complex C3b-BB. One of the end product of this pathway, the conversion of C3, is the first step in this pathway. In plasma, native C3 is not stable. Water is able to hydrolyze a Thioester bond and thus spontaneously activates a small number of these molecules. Once activated, it can bind to factor B, which has a molecular weight of 93,000 and is fairly abundant in the serum, at a level of 200 microgram per milliliter. Binding of C3b, sometimes called IC3, to B causes a conformational changes that makes B more susceptible to cleavage by serine protease. Thus, only bound factor B can be cleaved by factor D. The role of factor B is thus analogous to that of C2 in the classical pathway, because it forms an integral part of a C3 convertase. Factor B is a plasma protein that goes through a conformational changes when it binds to factor B. It is a serine protease with a molecular weight of 24,000 and its only substrate is bound factor B. When bound to factor B, the catalytic site on factor D opens, making it an active enzyme. The concentration of factor D in the plasma is the lowest of all the complement proteins.